Welcome to Lecture 8 on the topic of photosynthesis, where we will concentrate on the carbon reactions. This lecture is separated into two parts, and this is the second part. Please ensure that you have watched the first part before continuing with this lecture. This lecture is part of the subject Plant Physiology, which is a component of the Bachelor of Agriculture and Technology, a degree that is offered jointly by both Melbourne Polytechnic and La Trobe University. My name is Dr. Nikki Cooley. Lecture 8 on the topic of photosynthesis, the carbon reactions, part 2, is part of a series of lectures offered in this subject. Please ensure that you follow the order of the lectures starting from lecture 1. Ensure that you have watched all the lectures before you continue on with this lecture, as some of the concepts covered in previous lectures are required for this lecture. In part 2, we are going to explore other metabolic pathways for the carbon reactions. These include the C4 plants, which undertake the Kratz anatomy, and an introduction to the Crassulacean acid metabolism, or CAM plants for short. I'd like you to watch the following video on cycle counting. This video will explain how you count carbon molecules and how you count the exchange of energy during the C3 carbon metabolism process. As stated in the previous lecture, you can count based on the production of one molecule compound, or you can count on the production of one p-gal molecule. Both methods are commonly used and both are correct, and this video will explain these concepts in further details. So let us review for a moment. Ribsco is an important enzyme used in the regeneration of the carbon-Benson cycle, and this was covered in part 1. The role of this enzyme is to catalyze RUBP. This is an essential part of the regeneration phase. Sometimes this does not always happen in plants, and there are problems in the regeneration phase. This problem with Ribsco is a very important problem in agriculture, and so we are going to spend a little bit of time trying to understand this. In order to understand the problems with Ribsco, it is important that you understand a little bit about evolution. Plants and the carbon-Benson cycle evolved within the Earth's atmosphere at a time when there was no oxygen present. It turns out that Ribsco can bind more readily to oxygen than it does to carbon dioxide if both are in the facility. So why is this a problem, and how does it occur? In order to explain the mechanisms we are going to show you here, we need to review what we have learned so far. Carbon dioxide is a molecule found in the atmosphere. It dissolves through the stomata pores into the chloroplast. Carbon dioxide is utilized in the stroma with the aid of energy that is produced from the light reaction. The light reaction occurs in the thylakoids. When stomata are open, transpiration or pull in many situations can occur, and this process of carbon dioxide entering into the stroma and being utilized in the carbon-Benson cycle is an efficient mechanism. But what happens when the stomata are closed? When the stomata are closed, no more new carbon dioxide can enter the stroma. Not only does carbon dioxide occur in the stroma, but also oxygen can diffuse from the atmosphere and is commonly found in the stroma. This can cause an issue, as oxygen is capable of binding to the enzyme Ribsco, the same as carbon dioxide is capable of binding Ribsco. If oxygen combines to the Ribsco enzyme, it is no longer able to function Not only can oxygen bind with Ribsco, but it is actually more favourable from a chemical perspective. That is, if you had an oxygen molecule present and a carbon dioxide molecule present, the Ribsco would bind onto the oxygen preferentially over carbon dioxide. So let us review this reaction. If stomata are open, an atmospheric CO2 can enter the leaves and go into the stroma. Carboxylation yields 2 molecules of the 3-phosphoglycerate. However, if the stomata are closed and a build-up of oxygen occurs in the stroma, different compounds are yielded. 1 molecule of phosphoglycerate is yielded, as well as 1 molecule of 2-phosphoglycerate. This is not used in the Calvin-Benson cycle. Oxygenation involves reactions in the chloroplast, leaf peroxisomes and mitochondria. This process is also known as photorespiration, and it causes partial loss of the production of on the fixation of carbon dioxide and the uptake of oxygen. These competing reactions have negative impacts on plant growth, and these also have negative impacts on productivity, yield, and can be seen often in agriculture, especially agriculture occurring in Australia. So the CO2 and the oxygen compete for Ribsco with RuBP. This is known as the carbon-2 oxidative photosynthesis. Oxygen creates 2-phosphoglycolate and 3-phosphoglycerate. 2-phosphoglycate is then metabolized by 2 other organelles, the perisomes and the mitochondria. In the recommended textbook, Figure 8.8 describes the process about what happens in these 2 reactions. So let us review. The balance between the Calvin-Benson cycle and the C2 cycle is determined by 3 factors. The first is inherent to the plant. The second is temperature, usually higher temperature, causing a tendency towards the C2 cycle rather than the Calvin-Benson cycle. And thirdly, the concentrations of the substrates, carbon dioxide and oxygen, in the stroma. If we look at increases in temperature, this modifies the kinetic constraints of Ribsco, increasing the rate of oxygenation. Temperature also lowers the concentration of carbon dioxide, more than that of oxygen in a solution in equilibrium with air. So you can begin to see that a progressive increase in temperature tilts the balance away from the Calvin-Benson cycle towards the C2 oxidative photosynthetic cycle. The adaptive value of photorespiration is a matter of debate. Firstly, it is thought to promote the recovery of carbon that would otherwise be lost as a 2-phosphoglycolate. Secondly, the formation of 3-phosphoglycerate prevents the lethal accumulation of glycerate in dead-end pathways. Thirdly, thought to minimize photoinhibition caused by excess reductant formed in chloroplasts under stress, for example high illumination, high temperature and carbon dioxide or water deficit. And lastly, the major source of hydrogen peroxide is photorespiration. Although toxic, hydrogen peroxide also acts as a major signaling molecule and regulates the cell homeostasis, and thus has a positive role. Let us talk about engineering of photorespiration. Efficient ways to increase biomass are being sought as solutions to the global food and energy shortages. Can land plants be adapted to enhance photosynthesis? Well, enhancing photosynthetic efficiency or reducing photorespiration should improve yields of crops such as wheat, rice and cotton. Attempts to modify the performance of Ribsco, unsuccessful to date. Plants are generally stunted and die under atmospheric CO2. Alternative pathways for retrieving carbon of 2-phosphoglycolate has been introduced into chloroplasts. These are via bacteria E. coli, glycocarbotic pathways. This lowers the flux of metabolites through peroxides and mitochondria, thus avoiding the use of ATP. To date in our lectures on photosynthesis, the carbon reaction, and on photosynthesis, the light reaction, we have only explored one type of carbon metabolism process. The plants that exhibit this kind of metabolism are described as the C3 plants. But what if there was another way? In the next part of this lecture, we're going to explore some other types of metabolism. C4 plants have evolved anatopically to be different from C3 plants. These differences allow the plant to compartmentalize the carbon fixation part of photosynthesis. So why do they do this? Well, to minimize the oxygenase activity of Ribsco and loss of carbon through photorespiration. C4 photosynthesis evolved to compensate for limitations of low carbon dioxide. In the 1950s, C4 photosynthesis was discovered in sugarcane and maize. First stable products are the 4-carbon malate and aspartate. It takes place in two cell types called mesophyll and bundle sheath cells. The enzyme phosphophenylpyruvate carboxylase, or PPCase for short, catalyzes the carboxylation rather than Ribsco. The metabolism in the C4 plants, such as sugarcane, is called the C4 carbon cycle. The resulting 4-carbon acid flows across the diffusion barrier to the vascular region and is decarboxylated, releasing carbon dioxide. This carbon dioxide is refixed by Ribsco via the Calvin-Benson cycle. The C4 cycle is associated with a particular leaf anatomy, and this is called the Krebs anatomy. An inner ring of the bundle sheath cells around the vascular tissue and an outer layer of mesophyll in close contact with the epidermis describes this anatomy. This anatomy enables the compartmentalization of enzymes for C4 in two different types of cells. So, as in many things in plant physiology, the structure is very linked to the mechanism. Here we have a C4 anatomy. This was first described after the scientist Krebs, who called it Krebs anatomy. You will note from the mouse the area of the bundle sheath cells. This is around the vascular tissues and an outer layer that is in close contact with the epidermis. C4 plants have evolved to use a compound called PEP, and this is used to fix carbon dioxide. The evolutionary advantage of the development of PEP is that it does not bind to oxygen, unlike RUBP. So in these plants, oxygen is no longer a problem. So let us follow in detail the 5 stages of the C4 transport of carbon dioxide. The first stage is the fixation of the hydrogen carbonate through carboxylation of the phosphophenylpyruvate, or PEP, by the enzyme PPCase. This occurs in the mesophyll cells. Oxalate is converted into malate or aspartate. The second stage is the transport of the 4-carbon malate to the bundle sheath cells. The third stage is decarboxylation of malate into carbon dioxide and pyruvate. Carbon dioxide enters the chloroplast and the Calvin-Benson cycle. In the fourth stage, the 3-carbon pyruvate is transported back into the mesophyll. And the final stage is the 5th stage, where regeneration of the hydrogen carbonate acceptor pyruvate is converted back to phosphophenol-pyruvate. The formula on your slide shows the chemical reactions. So let us look at that formula in detail. Pyruvate plus Pi, which stands for inorganic phosphate, plus adenosine triphosphate, or ATP, will give you three products: phosphophenol-pyruvate plus adenosine diphosphate, or ADP, plus two inorganic phosphates. The oxalate is a C4 compound, and it is this compound that the cycle is named after, hence the name C4. It is the first intermediate made in the cycle. Two cells are involved in one metabolic pathway, and it may be easier to demonstrate the pathway with an illustration that shows both the metabolic processes and the role of the anatomy. On the slide you will see an illustration of the C4 pathway going through anatomical features. Using the mouse I will demonstrate the pathway. The carbon dioxide molecule is absorbed into the mesophyll cells. The carbon dioxide molecules will bind to the PEP carboxylase, and this will form the C4 compound the pathway is named after. Firstly, you will note that this part of the pathway is different from the C3 cycle. As in the C3 cycle, the compound that carbon dioxide binds to is RuBP. The reason that PP is selected as a preferred compound is that it does not bind to oxygen. Oxygen, like in C3 plants, is found in the mesophyll cells too. Once the C4 compound is formed, this molecule will move through the membrane into the bundle sheath cells. Here, the carbon dioxide will be released from that C4 compound and it binds to Ribsco. And this is where the cycle is the same as the C3 plants. The 3PG compound is produced through a number of stages that aren't illustrated on this slide, and you have glucose, your molecule sugar, produced in the vascular tissue. Regeneration, as in the C3 cycle, occurs with RuBP and ATP from energy. And so this demonstrates how structure and mechanism work very well together to give you the C4 pathway. I would like to recommend that you watch the following YouTube video that is on the topic of C4 and CAM plants. This will help with your understanding around this topic. Please make notes on the mechanism of C4 CAM and the differences while you are watching this video, and add these to your lecture notes here. The C4 cycle is known to occur in 18 families of monocots and dicots, particularly in the Poaceae. That includes corn, millet, sorghum and sugarcane. The transport processes facilitated by the plasma desmata generates a much higher concentration of carbon dioxide in the bundle sheath cells, thus suppressing oxygen and photorespiration. And these are the advantages of the development of this system. The bundle sheath and the mesophyll cells show large differences in enzyme composition. For example, PEPKs in the mesophyll, Ribsco in the bundle sheath, NADP malic enzyme in the mitochondria, and PEPKAs in the cytosol. Starch accumulates mainly in the chloroplasts of the bundle sheath. So let's talk about the C4 cycle and temperature. Elevated temperature decreases both the capacity of Ribsco and the solubility of carbon dioxide. In C4 plants, two features reduce these effects. The first feature is a high affinity of PEPAs for its substrate saturates the enzyme as a reduced carbon dioxide concentrations. This in turn enables C4 plants to reduce stomatal aperture, conserving water while fixing carbon dioxide at rates greater than C3 plants. The high concentration of carbon dioxide in the bundle sheath cells minimizes C2 photorespiration, both significant advantages. The C4 cycle is known to occur in 18 families of monocots and dicots, particularly in the Poaceae. That includes corn, millet, sorghum and sugarcane. The transport processes facilitated by the plasma desmata generates a much higher concentration of carbon dioxide in the bundle sheath cells, thus suppressing oxygen and photorespiration. And these are the advantages of the development of this system. The bundle sheath and the mesophyll cells show large differences in enzyme composition. For example, PEPKs in the mesophyll, Ribsco in the bundle sheath, NADP malic enzyme in the mitochondria, and PEPKAs in the cytosol. Starch accumulates mainly in the chloroplasts of the bundle sheath. So to bring everything together on C4 carbon metabolism, elevated temperature decreases both the capacity of Ribsco and the solubility of carbon dioxide. C4 plants have adapted two features to reduce these effects. One, what is called a high affinity of PEPKs for its substrate saturates the enzyme at reduced CO2 concentrations. This enables the C4 plants to reduce the stomatal aperture and then conserving water, while still being able to fix CO2 at rates that are far greater than C3 plants in these situations. The higher concentrations of carbon dioxide in the bundle sheath cells minimizes C2 photorespiration. So now we're going to move away from C4 metabolism and look at another class of metabolism. That is of the Crassulacean acid metabolism, or CAM for short. I prefer to refer these plants as CAM as it's much easier to say than the full terms. CAM plants have evolved another way of fixing carbon in arid, dry conditions. Some examples of CAM plants include pineapple, agave, cacti and orchids, and they all exhibit another mechanism for concentrating carbon dioxide at the site of Ribsco. An important attribute of CAM photosynthesis is the plant's capacity to attain high biomass in what is considered otherwise to be hostile, arid, hot, dry environments. Anatomical features that minimize water loss can include thick cuticles, large vacuoles, and stomata with very small and sometimes embedded apertures, and the tight packing of mesophyll cells. Typically, a CAM plant loses 50 to 100 grams of water for every gram of carbon dioxide that is gained or fixed. For the same amount of fixation in C4 plants, 250 to 300 grams is lost, and this number is even greater in C3 plants at 400 to 500 grams of water. This shows you how efficient CAM plants have become. So how do the CAM plants do it? Well, it's quite simple really. If you lose water through stomata during the day, why not close them? And in fact, that's exactly what the CAM plants do. This is the difference between the C3 and the C4 plants, as they will not close their stomata, and only close their stomata in specific climate conditions, while CAM plants close their stomata all of the time. Obviously, this reduces water loss, but it would limit carbon dioxide, wouldn't it? Well, CAM plants have adapted this so that they can still photosynthesize and keep their stomata closed during the day. During the night, this is the time when CAM plants will collect their carbon dioxide. This diagram sums the overall process of CAM carbon fixation. On the top we have a picture of the moon indicating the night. During the night, the stomata are open. CO2 is allowed into the plant and carbon fixation occurs by the process of PEP, You will remember this from the C4 plants, and it is identical. The fixation of the CO2 is stored as the compound malate, also known as malic acid. Then the sun comes up in the morning. The plant has a large concentration of malate, and this can be converted to allow the carbon dioxide to be available for the C3 RUBP Calvin-Benson cycle. In CAM plants, the initial capture of carbon dioxide and the final incorporation of carbohydrates can occur almost 12 hours apart over the 24-hour light-dark cycle. This figure from the Plant Physiology Recommended Textbook sums up the processes. In the dark, as we stated before, the stomata are open. Atmospheric CO2 is absorbed into the cytosol. This is where it is able to be combined with the compound PEP carboxylase. There are a number of steps from the production of PEP carboxylase to malic acid, which is then stored with its important CO2 combined in the vacuole. This process has a regeneration stage. While during the light hours, stomata are closed, as you can see from the right-hand image. In the cytosol, malic acid is released from the vacuole as malate and then is transferred into the chloroplast, where the CO2 is used in the Calvin-Benson cycle for the production of glucose and then starch. CAM plants have a 24-hour cycle, and this is divided into 4 stages. Stage 1 is the nocturnal phase. Stomata are open and CO2 is captured and stored in the vacuoles as maleate. Stage 2, early morning. Transient phase, allowing a shift in the metabolism to prepare for phase 3. Daytime is when phase 3 occurs. Stomata are closed, leaves are photosynthesizing. Stored malate is decarboxylated, resulting in high concentrations of carbon dioxide around Ribsco, thus minimizing photorespiration and also water loss. And the final phase, phase 4, late afternoon, transient phase to prepare for the phase 1 activity of Ribsco decreases while activity of PEPKs increases. In part 1 of the lecture on photosynthesis, you were asked to read the Tays and Zeiger chapter on plant physiology, chapter 8, Photosynthesis, the carbon reactions. Now that you have listened to this lecture and begin to understand the differences between the C3, C4 and CAM, I'd like you to review that and insert any additional notes that you feel will complement this topic in this section in your notes. So let's summarize all that we have learnt in this lecture 8, photosynthesis, on the carbon reactions. We started by learning about the compound RUBP and its important role in C3 metabolism. We learnt about its ability to fix carbon dioxide, but also about the relationship with oxygen, and this can result in inefficiencies in C3 carbon metabolism processes. These inefficiencies have led to other carbon metabolism processes that we learnt about. These include the C4 and CAM metabolism. After you have finished this lecture, watched the associated videos and conducted the recommended reading from Tays and Zeiger, I'd like you to think about the similarities and the differences of all of these mechanisms and learn the important components of each. What is the role that photorespiration has played, i.e. the role of oxygen, and how it has evolved adaptation in carbon metabolism. This brings us to the end of part 2 of this lecture.